Retrotransposons. So the human genome has multiple copies of retrotransposons, and these have been earmarked as repetitive elements that have contributed to the genome organization. So let us look at the learning outcomes of this session. Retrotransposons are class 1 type of transposable elements, which are further classified into mainly long terminal repeat retrotransposons and non-long terminal repeat retrotransposons. The long terminal repeat retrotransposons are like retroviruses. In fact, many of them are actually endogenous retroviruses, and they are viral-like in structure, retroviral-like in structure and mechanism. And of course, in this case, RNA is an intermediate. So from the RNA, complementary DNA is formed, and that complementary DNA gets integrated into a target site or into another part of the genome. And therefore, it follows what is called as the copy and paste mechanism. Non-LTR retrotransposons comprise the long interspersed nuclear elements, the short interspersed nuclear elements, and these are mostly target primed retrotransposons. These two follow the copy and paste mechanism. These transposable elements have been able to multiply in higher eukaryotic genomes, giving rise to repetitive DNA, and they have contributed to the evolution of genomes and also has been found to be associated with diseases. Now let us go to the long terminal repeat retrotransposons and look at its structure. So as the word suggests, a long terminal retrotransposon has an open reading frame, and this open reading frame comprises genes that give rise to proteins that are required for transposition, and flanking the open reading frame are long terminal repeats. On either side of these ORF are present long terminal repeats. What you observe from this point to this point is actually the transposable element, and these are what are called as target site duplication target site that are duplicated. So duplicate target sites, and these duplicate target sites are actually formed due to the process of transposition, and we will learn about it a little later. Now there are, so this is a general structure of the LTR retrotransposon, and within this LTR retrotransposon again are several families, and some of them, as mentioned earlier, are what we know as endogenous retroviruses. So for example, you have a lot of you have a family of what is called as Type 1 copia and Type 3 gypsy types, and because they are endogenous retroviral structures, you can see that the open reading frame also has genes for GAG and POL, and these GAG are basically genes that give rise to the coat proteins of the virus. And in terms of the genes that are responsible for giving rise to enzymes that are involved in the transposition, you have proteases, you have integrases, and you have reverse transcriptase. And flanking both the sides of this, you have what is termed as the long terminal repeats. Now you have for all elements a region in the 5' untranslated region. So this portion can be translated. This is the untranslated region. Within this untranslated region is present primer binding sequence or primer binding site, which we will look at what the function is later on. And at the other end, you have what is called as the polypyrimidine tract. So this is also of use again during transposition itself. So you see the same kind of thing in the metavirus family of retrotransposon, but the only difference being is the order in which the enzyme genes are present. So therefore, this is an overall structure that is there for the long terminal repeat retrotransposons. Now let us look at the life cycle, therefore, of the long terminal repeat retrotransposons. So within the nucleus, you have these transposable elements getting transcribed by RNA polymerases, and the mRNA that is formed inside the nucleus is translocated into the cytoplasm, and once it is in the cytoplasm, the mRNA that is obtained from the open reading frames present in the transposon are translated into its products. So you get the GAG protein, you get the reverse transcriptase, you get the RNase H, you get the integrase. All of these proteins that are there are formed in the cytosol, and once formed, they assemble into what is called as the viral-like um, uh, particles. So they're not really viruses, but they are viral-like particles. And within the viral-like particles are present the mRNA, which is then reverse transcribed by the reverse transcriptase to give rise to cDNA. And this cDNA is a template to form the second strand to form a double-stranded DNA. Now, this double-stranded complementary DNA it binds to the integrase and is taken inside the nucleus again. And once inside the nucleus, you have this complementary DNA that is formed, incorporated into another chromosome. So you can see that it originated from this chromosome, but once the cycle is over, you see that it has got incorporated into another genome. So that basically means that you had a transposon here. Now, because um, of the mechanism of copy and paste, so your copy has been formed and it is pasted at another site. So now you have two copies of the same present in the genome of this particular cell. So this is how you can see how you have the same element being repeated several times in the genome. So you will have multiple copies of the same transposon present within a genome because of its copy and paste mechanism. Now, what has been, of course, observed is that in many cells, including human cells, you have this copy and paste mechanism getting regulated. So there are mechanisms that the cells have adopted to silence this copying and pasting mechanism if needed. Or what has been observed is that you have means of regulating so that not too many copies are formed. Or if there is no regulation, then many copies will be formed, and the pasting of these copies into a particular target site or into a particular site in another chromosome may lead to disruption of the gene in which they have got incorporated. So these are things that have been observed with respect to the retrotransposon per se. Now let us look at the mechanism of these viral-like retrotransposons. So, as we have seen, this is the basic structure of the LTR retrotransposon, and the promoter lies within the LTR region. So, you have the RNA pol coming and binding to the promoter region and transcribing the transposon element, and so you get the element RNA. Now, this element RNA is then reverse transcribed by the reverse transcriptase. So, the reverse transcriptase actually has dual activity of endonuclease as well as polymerase activity. So, it is able to use the RNA as a template and synthesize the complementary DNA. And this complementary DNA then is integrated within a target DNA. So, for it to be integrated into the target DNA, it is the integrase enzyme that catalyzes first the 3' end cleavage of the transposon. So, a dinucleotide is released from both the ends to create a staggered end, a very small staggered end, but the 3' OH of this same is going to carry out a nucleophilic attack on a target DNA. So both the 3' OH is going to do so, okay, and then with the help of the integrase, this transposon gets integrated such that the target DNA has a small region which has a gap because of creating again a staggered cut in the target DNA as well. So you can see this cuts at this position and this cuts at this position. So you can see that there is a staggered, it's not exactly at the same point, because of which what happens therefore is once the complementary DNA is integrated, you have a portion of the target DNA that needs to be filled up. So there is a gap on both the sides, and that is again filled up, and it's generally the DNA repair proteins that fill up this gap, and then using DNA ligase, all these ends are sealed. So what you now see is that this transposon, which is in one chromosome, is now present in another chromosome or another DNA, and you can see how you have target site duplication, because you can see that this portion and this portion of the target DNA is repeated. So these blue repeats that you see is the target site duplication that has happened. Now let us actually try to look at how this reverse transcription is carried out by some of these retrotransposons, and it is in actuality quite a complex procedure, and also many things about it is still not very clear, and yet we will get a kind of understanding of how this reverse transcription is carried out. So this is basically the RNA element that has formed from the retrotransposon, and this RNA element on both the sides has a repeat which is similar, okay, which is the same actually, and at the 5' end of the RNA you have what is the 5' UTR, so U5, and you also have the 3' UTR. So UTR is basically the untranslated region of the RNA, and now within this of course you have the open reading frame. So at the 5' UTR is present a sequence that is called as primer binding sequence. That primer binding sequence is bound to by the 3' end of a tRNA, and this 3' end on binding to the primer binding sequence, this 3' end of the tRNA is actually acting as a primer. So the reverse transcriptase uses this 3' OH and synthesizes the DNA. This is the complementary DNA. So synthesizes the complementary DNA, and you have the U5 and the R of this region being synthesized. Now what happens is very interesting. This entire complex of the DNA with the tRNA jumps to the other end, which is the 3' end of the RNA element of the transposon, and because the R regions are the same, this is complementary to this R, so this will be complementary to this R as well. So this R portion comes and binds to this R element or R region of the element. And this now functions as the 3' OH, which is used by the reverse transcriptase to transcribe the entire RNA element, including the U5 and R all over again. So this is the first strand of the complementary DNA that is formed. Now, what happens is that the ORF also gives rise to RNase H. So that RNase H is going to cut the RNA that was present. So you see that you have the RNA present over here, correct? This is the original RNA element, and this original RNA element is going to be degraded by the RNase H. But a small portion of that RNA remains bound to the polypyrimidine tract that is present in the 3' UTR. So using this, the reverse transcriptase now transcribes this portion. Okay, since it uses the 3' OH end, and it will transcribe the U3, the R, and the U5 again. And you can also see that because of the endonuclease activity, it has removed the tRNA from here. So what you have now is a strand that contains the U3, R, and the U5. So the second jump now that happens is that this R and the U5 can bind to this R and the U5 in the first complementary DNA strand. Because it can jump and bind over here, this becomes the primer for the formation of the second complementary DNA strands to give finally the complete DNA, and this definitely is the complementary DNA. Also, what happens is that you have the LTR regions formed again because of the U3R and U5 being formed on both the sides. So this therefore is the long terminal repeat retrotransposon that has been converted into a complementary DNA. Okay, and this complementary DNA is then integrated into the target DNA. So this is how the reverse transcription process happens in by the LTR retrotransposons. Now let us go to the next type of the retrotransposons, and that is the non-LTR, that is long terminal repeat retrotransposons. So what you have in this retrotransposons, very obvious, is that you don't have the LTRs flanking the transposon ORF element. You do have the 5' UTR. You have two ORFs, okay, ORF1 and ORF2, which will give rise to the products ORF1 and ORF2. You have the 3' UTR, but interestingly, what is present is a poly A present at one end of the non-LTR retrotransposon. Because of the presence of this poly A, these non-LTR retrotransposons are also named as poly A retrotransposons, and these retrotransposons generally carry out target site primed reverse transcription. That means very specific targets are primed, and then after priming they carry out reverse transcription. So unlike in the LTR, where you have a complete complementary DNA formed and then that gets integrated, here what happens is that the RNA that is formed from this transposon goes and binds to the target, and then you have the complementary DNA being formed. So let us just look at the mechanism. So within the nucleus, this transposon is transcribed. Okay, on transcription, you will get the line mRNA with the 5' UTR, 3' UTR, and the poly A tail. And this line mRNA is transported out from the nucleus into the cytoplasm. And once into the cytoplasm, the ORF1, ORF2 proteins are translated, and these ORF1 and ORF2 proteins then again go and bind to the same line mRNA. So this complex of RNA protein complex is then transported to the nucleus again. So what began in the nucleus goes to the cytosol and then goes back to the nucleus again, so that this gets into a target site. Now, such transposons are what we know as long interspersed nuclear elements. Okay, and you have several examples of long interspersed nuclear elements. For example, L1 is one of the types which has been, in fact, implicated in several diseases like colorectal cancer. Now, let's look at what happens after you have the line mRNA with the protein complex get into the nucleus. So once it comes into the nucleus, the poly A tail specifically looks for a target where you have poly T. So this poly A tail with the ORF will need to come and bind to the poly T. So therefore what is done is the ORF makes a cut in this strand, okay, and having made the cut, the poly A tail of the line mRNA binds to the poly T, as you can see over here. Because the cut has been formed, the target DNA, the 3' is exposed over here, and using this 3', the ORF2, which has both DNA endonuclease activity as well as reverse transcriptase activity, will use the RNA as a template and synthesize the complementary DNA. So you can very well see over here, once the poly A tail primes or is bound to the target, only then the complementary DNA is formed. So the T-rich sequence, as we mentioned, are the preferred sites of cleavage So the ORF2 itself has a DNA endonuclease activity. So that is why it cuts at the end of the poly T, okay, and that poly T can base pair with the poly A of the line mRNA. The Nick, of course, created, as mentioned earlier, a 3' OH, which is used by the reverse transcriptase to synthesize the complementary DNA. So once you have the line complementary DNA formed, okay, so the first cDNA has been synthesized. Now you have to have the second cDNA being synthesized. So for the synthesis of the second strand, the cleavage has to be in this strand, because here this has already been taken care of. Now in the target DNA, the second cut has to be in this strand. So the cut can be either here or it can be this side of the primed site, right? And interestingly, what has been observed is, so it could be either or, it could be here or it could be here. So the cut could be either or, and if the cut is on this side, then you have target site deletion, okay? While if it is here, then you have target site duplication. Many studies have revealed that this is what happens. And once the cut has been made, this RNA is degraded by the RNase H, and now this complementary DNA is available for synthesis of the second strand. So you can see how the second complementary DNA strand is synthesized. This is what has been synthesized, and then of course you have the DNA joining taking place by the repair proteins in the host. So what you see now is the line DNA is present in the target DNA region. So it arose from a different site, made a copy of itself in the form of an RNA intermediate, and that RNA intermediate primed the target, and from the target site you have the complementary DNA form, and that complementary DNA is now the complete line DNA, but it is in another chromosome or another part of the genome. So this is essentially very important to note, that you have a copy of it being formed. So in fact, you can see that you have a duplicate copy of the line DNA being present. Now let us go to the second type of the non-LTR retrotransposon, and that is the short interspersed nuclear elements. Now interestingly, the short interspersed nuclear elements are non-autonomous transposons. When we say non-autonomous transposons, it means that it does not have the enzymes for transposition in its structure. So it has to rely on the enzymes for transposition from another transposon. So let us look at the structure of sign, and you must take note of the fact that in humans, ALU sequence, ALU sequence is one of the sign transposons present, and several copies of the ALU sequences have been found in the human genome. So as you can see, the sign structure has a box A, a box B, and it has a region which is homology to the partner line. When you say partner line, it means that it is going to use the reverse transcriptase and endonuclease from this line segment. And of course, like the line structure, this also has a poly T. Okay. Because of the presence of the box A and box B, so this is the kind of promoter that is recognized by the transcription factor 3 of the eukaryotes to bring in the RNA polymerase 3, and it is RNA polymerase 3 that carries out the transcription of the signs. So ultimately, you would have the sign element, and this poly T actually works as a transcriptional terminator. But also interestingly, therefore, you will have now a poly A present in this sign mRNA. And this sign RNA, and this sign RNA now, because of the presence of poly A, can target again very specific DNA. Wherever they find poly T in the DNA, they will go and bind at that position. So from this end on, using the reverse transcriptase and endonuclease from a line transposable element, a cut will be made wherever there is a poly T. So the poly A part of the sign will bind to the poly T, and using this RNA, reverse transcriptase will synthesize the complementary DNA. it will make a cut on the other strand, and that strand will lead to again integration, and this integration will allow the formation of the second strand. And you can see at both the ends of the sign you would have the target site duplication, because you when it is cut, it leads to staggered, just about 2 to 3 nucleotides staggering, and because of that you would have copies of the same 2 3 nucleotides present on both the sides of the sign. So this is how you have got the retrotransposition product, and you can see that sign from one side is now present on another side. But of course the original site is maintained because it's a copy of the sign transposable element that has got integrated into the other site in a DNA. So this is how you can see how the copy number of the retrotransposons are increasing. Similarly, there is another retrotransposon which is called as Penelope-like element, and this Penelope-like element also again uses a similar kind of mechanism to get incorporated, or to get to make copies and then get incorporated into the genome. So with this, let us make the conclusions. The retrotransposons are of majorly two types: non-LTR retrotransposons and LTR retrotransposons with RNA as an intermediate in the process of copy and paste mechanism. The LTR retrotransposons are retroviral-like in their mechanism, as they have an open reading frame flanked by long terminal repeats. Now, this ORF gives rise to proteins and enzymes that help in making a copy of the element and integrating it at another site. So, when we are talking about of the enzymes, we know there is an RNase H, there will be a reverse transcriptase, there would be an integrase. All of these are present in the ORF of the LTR retrotransposons. The non-LTR retrotransposons exhibit target primed reverse transcription, and the lines are autonomous non-LTR transposons that help the short interspersed nuclear elements. So that is also the case. So signs are non-autonomous non-LTR retrotransposons that use the trans-acting molecules originating from lines to carry out transposition. So lines can carry out transposition on their own, but signs need the help of lines to carry out transposition. So retrotransposons occupy a significant space in the eukaryotic genomes as they go about making their own copies and integrating these copies in different parts of the genome, influencing gene function and evolution. Thank you.